Zoom.
Okay.
So we don't want them to wait too long either. And for a matter of record, since this is being recorded, I'm Leslie Sturman with the University of Florida IFAS Nature Coast Biological Station, in case I forget. And I think we'll introduce the speakers as they come up for the sake of time. And just a brief overview, I think most clam growers will agree that your survival over the summer months tend to be a little bit lower than what you typically observe in winter months. That's considered to be a chronic mortality, particularly of those clams that are just getting ready to reach market size. But over the most recent years, we've seen increased significant mortalities. Even before Hurricane Idalia hit in late August, growers were reporting not just losses of market-sized clams, but even their nursery seed clams. So as a result of Hurricane Idalia, Florida Sea Grant had a little bit of funding, disaster funding, and Mike Allen and I wrote a proposal to start looking to address these summer mortalities of culture hard clams, and not just in Cedar Key, but it seems to be predominant here. And this is really a multi-step project to start to begin looking at those contributing factors to those mortalities. To get started, the first step was to ask for growers' input. You know, what kind of mortalities are you seeing? What do you think some of the possible causes are? And what are some of the needs you have to address this? And to do that, we worked with the Cedar Key Aquaculture Association. And this is not, sorry. And they helped draft some questions, and they sent out that survey in March of 2024. And Rose Cantwell, who's chair of the Cedar Key Aquaculture Association, she's going to be giving the results of that survey in just a few minutes. The next step was to invite researchers, extension specialists, and others from various institutions, UF, FAU, Gulf Shellfish Institute, in a roundtable session. We met the following month in April at the NCBS facility. We talked about some of the survey results, ongoing and current and future projects, as well as what researchers or expertise they could bring to the table. Now, the summary of that roundtable session is posted to the website there. We're not going to review that today. But as a result of that roundtable session and the little bit of funding we received from Florida Sea Grant, we had proposals requested of those participants to initiate almost immediately that summer any kind of preliminary research or monitoring that would be needed. And that's what we're going to do here today. We're going to report on these five projects that were funded and conducted during the summer of last year. The first is monitoring and understanding water quality parameters. I'll be giving that report. And that's followed by Todd Osborne, who is with the Water and Ecosystem Science Department at UF. And his work was on sediment organic matter as an indicator of summer mortality. Unfortunately, Todd has had a family health issue, and I'm going to try and give his report today. Susan Larimore from Florida Atlantic University, Harper Branch, is here today to talk about her project on health assessment of clams collected during the summer, last summer. Lisa's here in Cedar Key. Sarah Hutchins and Stephen Hestenberg with Gulf Shellfish Institute. Their project was assessing performance of thermally selected clams during grow out. And we'll finish up with Shirley Baker, again from the University of Florida Fisheries and Aquatic Science Program. And her work was more of a lab work, looking at thermal tolerances and physiological and behavioral responses of clams. All right, but before we get started on these reports and Rose's summary, I want all of you to take out your phone. And also, folks on Zoom, take out your phone and click on that QR code. And there will be a brief survey. I've already given that to. Oh, you don't have your phone?
It always rings when I bring it, so I decided not to bring it today.
But we decided to take it.
There's a button and imagine turning it on.
We really gonna pick on me for trying to be polite. It's unusual.
That's true.
Your hair looks nice today.
Oh my goodness.
See, he'll be nice.
We are recording this. So
Just call it.
Oh, okay. Thank you then. All right. So you click on the QR code. There's a brief questionnaire, 4 questions, I believe. All right, but not seeing any responses. So Do you, well, you're not gonna see responses on the yellow paper. Do you need to do that? Oh, I see it.
No, I.
You wanna do We'll collect these.
We shouldn't be filming out over this. Never mind.
All right.
I was just trying to participate.
Well, I appreciate that.
I see, yeah, because you're gonna
All right, if everybody's finished, we're gonna move on.
Should these be filming out or just the farmers?
I don't care. I think any one body that's gonna answer this is fine by me at this point in time.
Are you getting permission off the?
Yeah. Yeah. All right.
Good. 1 minute maybe?
I think everybody's done. We'll collect those yellow questionnaires later and add that in. Okay, let's move on.
Yeah.
And I think everybody on Zoom could also participate in that. You just don't get email. All right, I'm gonna start with me and that's.
Yes. You or
I have,
Rose?
huh?
Rose is going?
Oh, Rose is gone. Sorry, Rose. You're not getting out of this. Yes, Rose.
Like I'm there.
Yes, you have to come up here. This is a brief summary of that survey that went out in March, and if you just tell me when to move it forward, I'll do that for you. How's that? You're fine, you're fine. It's just gonna pick up around.
Oh, okay.
Okay.
Hi, I'm Rose, and I'm the chair of the Aquaculture Association, and basically, like Leslie said, we did a survey back in March to our growers. I think we sent out over 160, and we had maybe like a 25% response, which is actually pretty good considering us. So I was kind of impressed with it. Okay, so click, Leslie.
Oh, sorry.
All right, we just did that in March, moving on. Okay, we had asked, in this survey, again, we kind of tailored it with our group. And these are some of the questions, number of years you've done clam farming in Cedar Key. And it's pretty much everything I'm gonna show you is self-explanatory on there. I'm just gonna kind of walk us through it. The average was 19 years, which again, and if you notice, it was kind of 26 to 30 years. So we're still kind of with the old group still, but the new group is starting to come up, 1 to 5 years. So it's encouraging. So moving on, the number of leases you plant in Cedar Key. As you see, the average ended up being like 27% in 1 to 5 years, or 1 to 5 leases, I'm sorry. Moving on. Are clams growing faster now, slower, or about the same as when you first started farming? So we've got a little over 50% ratio on the about the same. Our clam mortalities from plant to harvest over the past 5 years, worse, better, again, this went to 2023 after Idalia. Worse, better, or unchanged from when we first started. And basically just kind of focus on the red. And as you can see where it is, it's gotten a lot worse. I mean, that's a pretty high percent according to our stats. Sorry.
Oh, he has a question.
Oh, of course he does.
Could you skip back?
Go back one less, please.
More?
This one?
One more.
No, go one more forward.
Oh, 4.
One there. Doesn't 22 and 23 get skewed because the hurricanes? That's not really that, how do we?
We didn't
How
have
do we-
hurricanes in 2022, but we certainly did in 2023.
And what year was for me?
For me was 17.
Was 2016. 2016.
So my question is, do we skew to 83% because there was an actual disaster in there? How do you guys?
That's a
how do
good
you
question,
incorporate
but
that with your thermal variance?
We can on that. We didn't ask to just, but we know, and
No,
the growers were reporting losses way before the end of August.
Yeah.
2023.
right.
Yeah.
Oh,
so I don't think it's
I
skewed.
just didn't know, I didn't know if that was incorporated into the
Yeah.
resolution. Okay, sorry, Bobby.
Ask all you want. Moving
right.
on. Okay, when, I think we all know the answer to this and expected what this answer with the percent of 95 what it would have been. Is it during what season are crop mortalities usually observed? Check all that apply, and obviously it's summer months. That's kind of the way it's been from day one. What sizes of clams are most vulnerable to mortality events? This is kind of interesting that it kind of was almost even a little bit, except for the sub-adults. Is that like raceways? Is that what?
Yeah,
Yeah, okay, that's a big reference.
Question,
Mm-hmm.
is that survey results or from growers?
That was from the 39 growers that responded.
this was sent to all certified Lee County plant growers.
I'm curious what it would be in research.
And what it would be what?
If it was research, what the numbers would show.
Well, if you research the same 39, it'd be the same one.
Well, a researcher versus a plant grower.
Oh, is that what you're asking? So I'm sorry.
I don't know how to answer. I'm sorry, yeah. All right,
What do you consider the top causes of coral mortalities? You could see that, I mean, obviously the higher one, we just go from the higher percent to lower. Salinity, temperature, oxygen, you can see all of them vary by sediments, food availability, disease, the fouling of the bags, predation, clam spawning, and sediment type. That was what was the we asked that question, and those are the responses that we got in that order.
Pretty good.
And again, don't think anybody was really surprised on all those answers based on what we know, all of the information that we have. At hand, moving on, Leslie. Does anybody have any questions on that?
this is the last one.
Oh,
I'm so
that's
sorry.
good.
No, you haven't gone over it.
Oh,
I moved it
so
forward.
you want to, okay, let's say.
Sorry.
What are the areas of research and monitoring would you like to see started this summer? So we went by, those are what we came up with on the left-hand side, and that's the percents we came with on the right-hand side. So obviously, everybody's mostly interested in the health assessment, genetic diversity, dissolved oxygen, which is kind of funny, salinity is further down, but that was our answer on the last one for the high one.
Yeah,
Carrying capacity, temperature, phytoplankton, environmental DNA, and soil sediments analysis. Same thing on that. That was higher up on the last survey question.
and I think that's it, Rose.
Okay. Anybody have any questions?
Is there all those ones that said what search projects are you gonna do for the summer? Has any action been taken on any of them?
Yes, and that's what we're gonna report on today. Not all of them were addressed. And actually, every researcher that submitted a proposal that was funded through Florida Sea Grant. It was preliminary work. They had to conduct that work during the summer of 2024. We got 2024, yeah, I can't keep these hurricanes straight, sorry. We got hit by Debbie, and then Elaine, and some of the work was curtailed over September and continued into October.
Oh,
All right, we're gonna move on. All right, and I'm gonna report on monitoring of water quality and trying to understand this due to climate change impacts that we are experiencing. I'm sorry, but I am getting over a cold. I'm not contagious, so I'm gonna cough every so often. All right, so I thought I'd start with looking at clam production. In order to get that annual data, the only way to get that data is through Department of Ag and Consumer Services. As you know, they require monthly reports from primary wholesalers and then aggregate that reports of purchasing of clams. They aggregate that data. We are looking at 2016 through 2024. The 7-year average between 2016 and 2022 was about 96 million clams. Whether this is right or not, it's what we have. It's the trends we're looking at. Then in 2023, we see a 12% decline in production, and followed by a 53% decline in production last year. These past 2 years have been devastating, creating challenges to our clam farmers. Concerns now about profitability and even sustainability in the face of climate change. So shellfish aquaculture, which is open water farming, is very vulnerable to the multiple drivers of climate change, and these include increased water temperatures, increased tropical storms and hurricanes, extreme variation in rainfall, sea level rise, and habitat loss. So the remaining slides, most of the slides on here are sources of data came from the University of Florida. We maintain sondes and loggers at our water quality monitoring stations and have done so for a number of years. NOAA has a tidal buoy right here providing real-time and archived data out of the dock. And then we also obtain reports from NOAA National Weather Service and National Hurricane Center and hydrological records through the Swanee River Water Management District. So the next three slides, we're looking at temperatures over a 17-year period, focusing on June here, in both 2023 and 2024. This little red dot is pretty small, like this. So water temperatures on the left, and this was at Dog Island. The dark black line is June of 2024. The red line is somewhat highlighted, is June of 2023. There's air temperatures from the upland station. And you can see in 2024, temperatures started rising quite fast, whereas it was much milder in 2023 for that month. Nonetheless, the air, and we used rankings from NOAA, the air temperatures were the third warmest in Levy County since their records of 1956. What we really wanted to tease out was the number of days in which water temperatures exceeded 90 degrees Fahrenheit. That's the threshold temperature at which above plants start showing signs of thermal stress, such as gaping, or inability to bury, and inability to feed. So you see for 2024, 7 days, only 1 day. In 2023, it was a much milder month. Moving on to July, again, black is the 2024 values. Red, if you can distinguish, didn't want to put this over a 17-year period. For 2023, air temperature, again, the third warmest since 1956 on record. You see temperatures in both years, 2023 and 24, is certainly increasing and exceeding 90 degrees. Look at the number of days exceeding 90 degrees Fahrenheit. 24 days last year, 21 days in 2023. Now looking at August, this heat wave continues in 2024 and 23. Well, actually, excuse me, in 2023, that heat wave continued. We do see milder temperatures in 2024, possibly in part because of Hurricane Debbie early in that month might have caused some relief in water temperatures. Air temperatures, the sixth warmest on record since 1956. Do you see that drop in temperatures reflected in the number of days exceeding 90 degrees down to 6, as opposed to 19 days in the prior year for that month. So these small little inexpensive temperature blockers here, again, we've distributed those quite a bit in years past. Some of you all have used them out at your lease. They're very good in describing temperature variability within and among lease areas. The temperatures I just showed you are from one fixed station. So that's good to better understand variability. And this is actually just a week from 2009 from 12 leases at Gulf Jackson, just showing that variability in temperatures. If you can see it, like the green line is much lower. And that could reflect differences in water depth, even soil properties or other factors contributing. Last summer, we distributed lockers to growers, both at Dog Island and Gulf Jackson. They were all lost due to Hurricane Helene. And this year, lockers are being made available as part of the statewide project. And Angela Collins, who's here today, has a few to distribute, or you can contact her there if you're interested. Right?
Mm-hmm. Yeah.
Good.
That's all.
Yes.
Yeah. It's okay.
The green line that's below the rest of them, is that, where was that log? Was it deeper? What was it, you know?
Well, I'm so glad you pointed that out. So now I have to explain that.
You're gonna get to it later. Wondered, like it would be informative if that's.
Actually, that's the monitoring station.
Oh, it is. That's the dock?
No, the monitoring station at the pipe of Gulf Jackson. And it's the SON sits 6 inches above the bottom. So it kind of is contradictory as to why. I'm not quite sure, but that is the temperature from that. And it is a different blocker. So might make a difference.
Jim's saying there's different currents there. Yeah, there's water.
You know where the pipe is.
Yeah.
Yeah.
By.
See, these are other factors that clam growers are intuitively, I know those differences.
Cool.
Yeah. Thank you.
All right,
Thank you, Tim.
Hey, Leslie, most of them know how to get in touch with me, but my phone number on there is wrong.
But I'm so sorry.
That's incorrect. That's okay.
I thought I went to your
Email's
email.
right, But it's just 941.
Oh, that's me.
That's okay.
I have to,
Just
I
say
have
it's right.
dyslexia type. Okay, certainly these hot temperatures we just reviewed have driven the past few years in hurricane season. Don't need to review this, but nonetheless, a trio of hurricanes that impacted our area over a 13-month period. Interesting research is associated with world weather attribution conclude with high confidence that Hurricane Helene was made worse by climate change. So now we're going to move on, and this slide is monthly totals of rainfall for June, July, and August from the years 2018 to 2024. There's plenty of stations in this county where you can get rainfall. We decided to report from the Cedar Key Forestry Tower, since that's what is used by our folks in DAX to monitor our shellfish harvesting areas. So what we did is impose over these monthly totals, an average, and this we got from Swanee River Water Management District. It's an average of that month over almost a century worth of data, 1927 to 2023. So then we also use rankings from Weather Service, rankings again of temperatures in the upland area, rainfall in the upland area in Lee County, and we're going to display those. So in June of 2023, almost 14 inches of rain, double that of the historical average, and the wettest on record. That's followed in July with less than 4 inches of rain, ranked the 17th driest over that century worth of data. And then again, in August of that same year, the third driest on record for months. This variation continues into 2024 with June, the 7th driest, and down in August we see 16 inches of rain almost, and ranked the 11th wettest over almost a century of data. So this variability in rainfall certainly leads to inconsistent salinities in our inshore coastal waters. And this is reflected, again, one of these graphs where we're plotting everything. This is of Gulf Jackson. This is a 14-year plot here. Again, dark black is 2024, and the red is 2023. And you see in June of 2023, we had a lot of rainfall. That's certainly reflected there with salinities falling below 10 during the month of July and into August before recovering. Because June and July was not as wet for 2024, we don't see those salinities falling into threshold values of 15 or 10 parts per thousand. But nonetheless, Gulf Jackson is also influenced by discharge of the Swanee River, so we certainly tend to see lower salinities on the west side of the island. And these lower temperatures below 15 parts, particularly in the hot summer months, add to water quality stressors of hard clams. In the winter, they tend to tolerate those lower salinities better.
Leslie?
Yes.
August,
Yes.
the black line from the 4th to the 22nd, what is it? I'm blind. Is that-
No, you're not. You're observant. So that was a test. No, we have to pull the loggers because of the hurricanes.
You what?
We pulled the loggers.
Oh, so-
Yeah, these are $20,000 piece of equipment. We moved those out in anticipation. Yeah. Yeah.
So both the Delia and Helene were in September. So is there any comparison that you have? I know you're trying to associate this with pre-hurricane conditions, but is there any comparison to rainfall after the hurricanes?
Well, those were monthly totals, so they certainly would influence, so that would incorporate the rainfall associated with those.
Only July and August, we should not see Chinese September.
I can get that data, I just don't have it right now to present to you. Yeah. But that's a good point, and I'll remember that. All right, and this is Dog Island again, less influenced by Swanee River, so we're not seeing those low drops in salinity. But again, in 2023, in the red line, we did see a brief drop below 15 parts. We're considering that to be a threshold stress level, in addition with high water temperatures. Okay, moving on. So according to NOAA, Cedar Key has the fourth fastest sea level rise acceleration in the US with almost 12 inches of rise over the past century. That's that graph on the left. And then on the bottom graph, you see extrapolation of observed sea level trends. That's the orange line here. It outpaces even the most extreme high scenario model. So land-based infrastructure that supports our clam growers, such as processing plants, are certainly beginning to move online. All right, moving on. Rapid degradation of our oyster reef habitat certainly has occurred over the past 30 years and documented in aerial photography with 88% loss of oyster reef habitat on offshore reefs. That declines as you move inshore, but still 50% loss. And these decadal changes are certainly very disturbing, as these reefs have existed for over 3,000 years. In addition, we're seeing loss of inshore coastal islands over a similar time frame. Again, you can look at aerial photography here, with Cedar Key now completely lost. I think in 2016, this top we had about, I don't know the percentage there, but that is completely gone now. And again, even more lost in the Clam Key. This certainly would alter the flow and discharge of the Swanee River that more influences the leases on the west coast of the island here. And growers do report variable salinities more recently, and certainly have reported unreliable plant production at these leases over the past few years. So last summer, we did put conductivity loggers, which measure temperature and salinity, both at the northeast and southeast corners of Dog Island. Here's the actual saw and monitoring station. We did put one at Pelican Reef at the only upright 6-inch PVC piling that exists there, and that was lost almost immediately. So let's look at the results of that. And we're just looking at July for those stations. Temperature was basically the same. We do see differences in salinity. Again, this is July of 2024, if you recall, the rainfall for June and July was pretty mild. So you're not seeing as much an influence on salinity, but nonetheless, we do see on an average for that month a 3 parts difference, parts per thousand difference. But I think what is more important, what you're seeing, is how often that salinity falls below that 15 parts per thousand kind of threshold, arbitrary threshold level that we did. Whereas at South Gulf Jackson, you're not seeing that at all. All right, so what's new? So we want to begin monitoring phytoplankton. That's your clam food. So in order to do so, we purchased sensors and added to our monitoring stations, to our sondes. They measure in fluorescence. So in order to convert that to chlorophyll A, which is the pigment in all plants, including phytoplankton, we've had to go out and take samples, process those for chlorophyll A, and then try and create a calibration curve. These are variable responses. And so this is the results of what's being monitored every 30 minutes at, I think this is the Dog Island sonde. In terms of fluorescent, little green dots are what we are measuring in terms of chlorophyll by processing. So we got a long ways to go due to that variability to get some sort of good fit. Now you could help with that. Every time you're out at Dog Island, we will give you a nice little bottle. And oops, we don't do that. And you can take orders and we can grab it at your shop. You just need to know the exact time that you took that so we can correlate that with the fluorescent reading on the sonde. All right, what else are we going to do? We start this month. We have put together a team, Shirley Baker, invertebrate physiologist, Ashley Smith, biogeochemist with UF, and we are going to work with a clam modeler from Virginia Institute of Marine Science, who has done a lot of work in Virginia waters on clam leases. And we're going to start to try and understand carrying capacity, ecosystem modeling, and carrying capacity of the water body supporting the Dog Island Oregon Reefs lease area. All right, so wrap this up. So certainly the challenges associated with climate weather related events have been recognized. There are now ongoing responses by industry, researchers, extension folks, but what we need continue and ongoing is resources support collaboration and innovation so we can sustain our industry's future. All right. There's a few questions. Let's do it now and then we're going to move on. Oh, gosh, thanks. All right, go ahead and open up the next. All right, no questions. We're going to move on. Angela said, was there questions on the chat? Okay. All right, as I mentioned earlier, unfortunately, Todd cannot be here, and we've worked with Todd on a number of projects. He's really gregarious, ongoing. It's really a shame he can't be here. Give his own presentation, might wake y'all up. But his project, what he looked at was sediment organic matter as an indicator of summer mortality. I might mention, obviously, Todd is a soil scientist. And he's now at the Whitney Lab, East Coast of Florida. All right, excuse me while I'm trying to go through this. I did bring this in because I wanted you to recognize some of the previous work he and fellow soil scientists have done working with you folks here in Cedar Key. And this was in 2007-8, where they did a spatial inventory of the Dog Island. This was the footprint of Dog Island way back then. That certainly has changed. And that map there is really just bathymetry or depth water that they were able to map out by going out and taking multiple samples all over them and getting, cussed at by growers. You're on my lease. Even when they weren't. But what I think is most interesting, they did all the soil properties, including sand content, clay content. This is soil organic matter content, which is the topic of this presentation. And I think some of you that know how production varies on, and I wish some more Dog Island producers were here, but I think it's looking at the output from organic matter and how you know production occurs on Dog Island. I think you might see a correlation, particularly with a high organic matter up here in the northeast corner. Of course, this is the sandbar, the spit that goes out, so that's high in sand, so you can see less organic matter.
Your highest producing lease is 805 to 807 to what is that, 820 all the way to the west.
Yeah, 825, 826.
Yes, that's your high.
I'm gonna tell you, I know how many times I help people do
Lease.
transfers of leases to 801.
Yeah, because
Some in this room. Okay, going on. All right, so this is actually Todd's presentation. As a way of background, high temperatures in summer are tending to increase hard clam mortality events. And his question was, could this be due to low oxygen, which is referred to as hypoxia, hydrogen sulfide toxicity, or a combination of both that are brought about through increased temperatures? So the presence of organic matter influences hypoxia by stimulating respiration in microorganisms, that's your bacteria and everything else that lives in that mud, including your clams, and can similarly fuel production of hydrogen sulfide. So under normal conditions, organic matter plus oxygen equals carbon dioxide, water, and energy. But when oxygen is absent, you end up with sulfide and hydrogen sulfide, which is not really good. All right, the methods he did, and some of you in the room participated in this, to test that hypothesis that sediment organic matter may contribute to summer mortality. We had growers participate, 3 leases at Dog Island and 3 at Gulf Jackson. And they sampled settlements monthly, June through August, not in September due to lean, and then again in October. They collected 6 replicate samples and they were taken alongside clam belts when they were harvested. So they went along as you harvested, they took samples. And then 3 replicate samples from an unfarmed area such as the easement there. They also reported estimates of survival from that plant that was harvested at the time of sampling. Then sediment organic matter content was measured in the lab. Those are the methods used. Then sediment organic matter water quality obtained from our sondes. And clam survivorship estimates obtained from growers were compared to investigate the role of sediment organic matter. I don't think you really can see that, but I think the results of his analysis of these data collected Well, first of all, there was a total of 189 soil samples that were tested. He used a regression analysis to look at observations of percent clam survival versus organic matter, and it didn't reveal a relationship between the variables. No. I'm going to comment on that later, but I want to go through his presentation first, and I think y'all might know some of the answers to this why there may not have been a correlation. All right, these are called box plots. Never seen them before. You go, what the heck's going on? I do that all the time. But they suggest the spread of the data. So we're looking at clam survival. Just look at this line. This is kind of an average. So this is based on survival estimates given by the growers who participated at Dog Island and Gulf Jackson. So obviously a higher survival coming from Dog Island reported by growers than the growers that participated in Gulf Jackson. I'm thinking I find that surprising. And then he compared that to organic matter at both the lease areas from the samples collected. It's the same. So that's again kind of showing that there's no relationship between the two. In fact, he said there was some positive correlation that might indicate that some sediment organic matter might be good. All right. What is he saying here? This is over the months that were sampled, and this is clam survival. Again, orange or pink, seeing Dog Island, Gulf Jackson being blue. Just look at the lines in these boxes. That just gives you the average. These are different plants, so it wasn't following necessarily this, so that's going to be variable. It does indicate that there might be different stressor conditions and responses. He did look at water quality data, but we're looking at July for Dog Island, and he used the same threshold values I do in reporting, 90 degrees Fahrenheit for temperature. He added dissolved oxygen concentration, which we also monitor continuously. He used 10 milligrams per liter or parts per million as a threshold value. And so the red lines indicate the threshold values for Dog Island in July. It illustrates a lack of direct correlation between temperature and hypoxia, that means low DO, and suggests additional drivers involved in these conditions. All right, sum it up. So sediment organic matter was not found to be a significant factor in hypoxia or mortality. Both of these leases had different survival over the summer. The hurricane disturbance had a net removal effect for this is what I want to touch base on for sediment organic matter, but marked negative impact on clams. Based on available data in July, dissolved oxygen was lowest at both sites, and hypoxia was most prominent. And water quality does not show direct temperature to DO relationships, suggesting additional variables, perhaps suspended organic matter, are influencing hypoxic conditions during excessive heat. It's almost a shame that we weren't able to take soil sediment samples for this study prior to Hurricane Idalia. I think you all know, none of us can think of anything positive from Idalia with the one exception. It cleaned the leases. Mm-hmm. And you all reported better survival of plants after Idalia. Terrestrial land farmers terrestrial farmers know that. They oxygenate their land in order to remove any kind of suspended matter, etc. So I think we did see some benefits to our soil quality after Hurricane Idalia, and thus affected really any kind of results from this one. But I think an important outtake from this presentation and understanding is that, over time, it might be either you have to let a certain portion of your lease remain fallow. They also did this study years ago too. And their results showed that for Dog Island, they recommended a fallow period of 2 to 3 months. That means you don't plant over that. And that would allow those soil properties to recover to those that are in the adjacent easement. And then for Gulf Jackson, it was longer. It was up to 4 to 6 weeks fallow time to again recover those soil properties to those on an unfarmed area. All right, well, we can't ask Tom any questions, and you certainly can't ask me because I don't know the answers.
Yeah.
So we're gonna move on, but yeah, Mike,
I'm noticing since Helene, not the stuff that was down during Helene, but my new stuff naturally, it's just seed mostly, but it's doing great. Just like it did after I did it. I think we're seeing the same thing after Helene, as you're talking about Vidalia.
yeah, I think these are definitely lessons learned. Yeah.
Yeah, even the survival bags from the storm, I mean, clams inside them, we're running up and stunning, right now they're taking
Taking
off.
Yeah.
Yeah.
They're growing more red.
So great out there, so red.
Yeah,
yeah.
You can take 2 fingers pop.
Yeah, but they're
Well,
growing.
that's good to.
No, if you can be cleaned out.
Well, I mean, it's good they grow, yeah, they just have. All right, we are gonna move on, and the next person, is there another?
So would you recommend that we like fill our bottom after we harvest, like on the street that option there?
Well, that's a question for you, that's a question for you.
Well, I'm asking you, research.
Well, I know with the limited amount of lease acreage that some of our larger scale growers have, and they have to keep replanting over and over, you might take that into consideration. Yeah,
Would it be legal to do?
yes,
Do it?
just don't ask. No, yes, no, yes, yeah, you can't, remember, you, yeah, Mike,
I have the luxury of.
You wanna
being
go ahead
a
and get
small
this?
farmer.
Oh, Andy has questions.
So I am able to rotate my crop on my lease to over a year before I put bags in the same spots. And I don't know that that's up to me any more than it is anybody else. And that's just me on my lease.
okay,
Did you hear what I said?
no, I'm looking at Andy Kane's
I
hand,
thought so.
wondering how long.
No, I said that I have the luxury of being able to leave my bottom.
Yes,
Time.
yes.
before I plant it.
yes,
A year or sometimes a little longer. And I don't think I'm seeing any more success survival wise and whatnot through all of this stuff we're dealing with than anybody else.
okay, all right,
Now, that's just 1-2 acre
Right.
plot.
right.
I'm seeing, for me, I'm seeing overstock bags. Actually, with higher survivability. Actually, could you actually only overstock bag every now and then?
Mhm.
I'm seeing, it's day and night difference between higher survivability. In overstock bag than I am with a
Wow,
Tell me you're not seeing it, so. What?
Are you seeing it?
Well,
Overstock bags.
I couldn't understand what he said.
He's saying about
That's it.
overstock bags.
USDA says.
Oh, that's survival. Well, I don't overstock my bags, so I wouldn't.
He said every now and then.
That's one thing, the state says there's a standard, no more than
this is
13
just
fifty
Andy,
bags.
guys.
And I pulled some bags that my guy. Last year, and I found that they were stocked at least 1,500, and I've got 1,400 claims coming out of each bag. And then I got claims that were bags that were stocked at 1,350, and I'm getting less than 800, and they're the same one.
Yeah, yeah.
It's just, I'm noticing that my overstock bags are having a higher survivability now. What that means, they're growing slower.
But they're
but
going, but it's supply density dependent.
I didn't see that. I've had some that were slightly overstock higher than others, but they
Yeah.
didn't seem to make any difference in my.
There's so much variability in
That's
a lot of
exactly
this, that is hard to conclude.
The
The
seed
management
quality,
practice
yeah.
that's gonna work across all leases and all seed sources. It's very hard. Listen, we do have one person on Zoom, that's Dr. Andy Kane, and he has his hand up. And is he put that in the chat?
Yep.
Can you read that?
Well, he's just gonna ask him, Mike.
I was just going to ask a quick question. Dissolved oxygen. Hey, everybody. Those dissolved oxygen concentrations that you just shared over time, those are off bottom. Those are water column measurements. Is that right, Leslie?
The sonds are located 4 to 6 inches off the bottom. We can't put them in the mud.
Right.
Okay,
Obviously,
so
having the bottom deal would be kind of cool too. But where it goes down below that line that you showed, right, where it dips really. So those are on the x-axis. Those are monthly. I can barely see it, but those are monthly notes, right? And so where it dips down below that line, is that like in the middle of the night and respiration, or is that in the middle of a day where it's hotter than?
I can't answer that question, but given the diurnal nature of dissolved oxygen, I think you would guess right that that probably is showing respiration overnight, and the lowest DOs tend to be right before sunrise. We have that data. I mean, I just have to go back and look and confirm that, but that diurnal variation is always dealing with, respiration at night is the lowest point. So yeah, more than likely, Andy.
Yeah,
All right.
all right, thanks.
Thank you, Andy. We're gonna move on. Susan, you ready? You tired of talking? Yeah. Nothing. You can just every so often try talking to that. I think that helps with, yes. Or, I find that to be so small. I'm just using that yardstick. I can just point.
All right, we got a few more people here than want to know. Good thing. Okay, for those of you who don't know me, my name is Susan Larimore, and I'm at the Harbor Branch campus on the other side of the state, Florida Atlantic University. My project was to look at 2 Cedar Key aquaculture zones, Dog Island and Gulf Jackson, and to determine what was causing issues, if there were any issues, with the health of clams during the summer, and to try to be able to relate them to either pathogenic organisms or water quality changes. Was there a difference between Dog Island and Gulf Jackson and the leases therein? And so to do that, we took monthly collections, as has been said with the other ones. September was a washout, so we've got June, July, August, and October. 20 clams per lease, 3 leases per site, and so that equated to about 120 clams per month that we looked at. We did a gross examination when we got the clams, where we took measurements of the clams. Then we just looked at them externally. Were they gaping or anything like that? We opened them up and looked at them internally, looked at the mantle condition, the meat condition, whether they had any lesions or anything else apparent that we could see. And then we took bacterial swabs of the mantle. We did 2 different types of bacteria that we looked at, total counts on marine auger. This is an example of that. And then Vibrio counts on the TCBS auger. If you've been to some of these workshops before, that's that green auger that we work with, and you'll see pictures of that. And then lastly, we took a cross section of the clams and we did a histological examination. We made slides of the tissue and looked at things like the digestive tract condition. Were they eating? Were there any pathogens inside? And tissue responses. This shows a hemocytic response and swelled tissue here. All those purple things are hemocytes or clam blood cells, basically.
You want to tell them what hemocytes are? That you just.
Yeah, clam blood cells.
Yeah.
And what that does in a clam, it carries both nutrition, but it also fights diseases. So it's a multi-purpose cell. Unlike what we have. So the first thing, of course, is measurements. Unfortunately, I don't have June's. We had a little bit of a communication difference as to who was doing the measurements. So I don't have a June baseline, but we do have July, August, and October. And you'll see a lot of these arrows. I like to use arrows and color code stoplights. So red, yellow, and green, you'll see. So July then becomes our baseline. And this is an average of the 3 leases at each site. So we did see, length is really what we like to look at when we're doing measurements, right? And we saw a drop off in length measurements from July to August, a lot more significant or drastic, I guess you could say, Gulf Jackson compared to Dog Island. Where you see these asterisks across the top, that means there's a difference between the 2 locations, Gulf Jackson and Dog Island. If you see something within it, that means there's a lease difference. You'll see that throughout as well. Then by October, things looked up and we had growth that was better than in July. In October for Gulf Jackson,
And it was roughly kind of a washout at Dog Island. Now, of course, these are 3 different leases that we're looking at here. And as Leslie had pointed out previously, these aren't from the same bags that these were taken from. So we're not following the same.
They're from the same grower and from the same lease, but as you all know, if you moved on to a different plant, it may be younger and you have a smaller size clam. And certainly that was the case in Gulf Jackson where all the older clams were dead. They picked younger clams, so
Yeah.
it is
Yeah.
a wash.
Yeah, kind of.
Get to the good stuff.
Get the good stuff. Okay, so looking at the gross clinical signs, this is before we open them up. We looked at things like shell abnormalities. I'm not showing that here because they were slight. You saw some chipping, you saw some deformed lips. In June and July, we saw some of the clams with brittle hinges, maybe 5 to 15%, but then it didn't continue through July and August. What we really noticed was this huge increase in gaping from June to July, and what I'm calling siphons out. Otherwise, the clam clamped down on because they were too weak to retract their siphon inside. And you can see, really, you went from 0 to 22 to 45%, depending on where you were located. And then in August, it got better. We're getting better, especially at Gulf Jackson. And then by October, the surviving clams, wonderful again.
Yes. If you recall, August of 2024 from the water quality data, that temperature was much milder. Yeah.
So then we opened them up and we looked at a lot of different things like if there was a difference in coloration of tissue. We looked for lesions, any type of pest or parasites. We didn't see anything like that in there at all. I thought mantle condition would be a good indicator of health perhaps. And this is what I mean by mantle retraction. You can see how the tissue wants to pull away and, you know, come inside the clam. They want to protect themselves. And you do see a lot of it, but it was very consistent throughout, and most of it was slight. This is a fairly moderate one, and you can see some swelling around here. So I didn't really see any changes in that from June and July. What we saw mostly was physiological condition, how fat the meat was. So in June, you've got, you know, 1/3 to almost half of your clams that were fat, looked really nice. And then you get to July, boom, especially at Gulf Jackson, you get this huge decrease. Slight decrease at Dog Island, and then look at August. So you've got this residual effect that just moves on into August that, wow, they're just all watery or in pretty poor condition. And then again, you start to see an increase in October. You know, again, we're back to what we were in June, or we're getting there. We're good.
Before you leave that slide.
Sure.
I mean, wouldn't you associate that? It's a shame that we don't have this chlorophyll data available, and we certainly want to create a database for that. But it's certainly, wouldn't you think that is also whether they physiologically can't feed, or is there not enough food in the water during those hot summer months?
Yeah, I mean, I think what would be really good for all of us different researchers that are looking at the sediment, chlorophyll, the health, to get together and try to pull all that together as one.
Yeah.
Because, you know,
They relate.
yeah, they relate. How they relate. We're kind of working on bits and pieces of the puzzle, and we kind of need to get together, throw it all together.
Isn't there a partnership between you and Dennis? On this, like a going coalition or something?
But an oyster coalition, but we certainly are gonna start working with the VIMS faculty member this month. Yeah.
Because I mean, that should help with what you're talking about.
Yes, it would, because they've been doing this much longer. Yeah, and have had the resources to do
Like
so.
I said, they can give resources.
Yeah.
Okay, so now we're gonna move to the bacteria. I said we took swabs of the mantle. Now, look at this disclaimer here. This is not the inside of the tissue. So this is basically what the clams are exposed to out in the wild, is what you can say. It's more what the bacterial counts are and numbers are in the water as much as it is the clams, because we're just looking at the mantle. We're not digging into.
So do y'all know what the mantle is?
Jill,
It's, no, can you tell me what the,
Okay, so the mantle surrounds the meat. It's that thin layer that surrounds the meat.
it's the skin of the clam. And it surrounds
Yeah,
all
in
of
the
the
water.
internal organs.
Thanks. Show you next time you open one up.
sounds good.
So if we take a look, this is just for the marine auger, so the total counts. This is a plate here. This was kind of interesting to me. So you actually have an increase in total counts starting in August at Gulf Jackson, and you have more bacteria in the water in October than you do in June or July. But here's the interesting part. Colony diversity. So this is a plate that has lots of different colonies on it. You can just by the color alone, you can tell that you got yellow, white, orange, red. So at least 4 different colonies here, colony types. In June, we had up to an average of 4 to 5 colony types per clam that we opened up. And this would signify how many clams had more than 4 colonies. So kind of looked a little bit more like this, although there may be one color that predominated. So you can see we had quite a bit. Now, when you get to July, look how everything drops. So our diversity out in the wild decreases. The bacterial diversity decreases. And then it goes up again in August. And then in October, it continues to go up and returns to what it was in June for Gulf Jackson, but for some reason at Dog Island, it takes a dive. No idea why. Now, just to point out, not all bacteria is bad bacteria. We tend to think bacteria is bad, but bacteria can be doing good things too, like cleaning the water, like converting ammonia to nitrite and nitrite to nitrogen. So actually cleaning the water up. In a healthy environment, diversity is a good thing. You want to have a lot of different types of bacteria in there, not just, 1 or 2.
I just wanted to say on your bacteria counts there. So keep in mind, bacteria tends to die faster in warmer temperatures, and bacteria lives longer in cooler temperatures. We see that in our fecal coliform counts taken by our shellfish guys. So.
Okay. Good comment. Vibrio. So this is the one you always worry about in aquaculture. I don't care whether you're, culturing fish or clams or shrimp. This is the one you worry about. And this was interesting too. So we had fairly low counts through the summer, some variation by site, but October, it really ramped up. However, the big story is the number of green versus yellow colonies. We tend to think of green as being more pathogenic or harmful. So these are the ones you have to worry about. And look at the big jump that you got from June to July in Dog Island. And then a little bit of delay at Gulf Jackson, but it jumped in August. And then boom, October, we're back to what we were, close to what we were in June. So just like the total bacterial diversity, you saw a change in the type of Vibrio that was out there as well. So how did this seem to affect clams internally? So these are the slide preparations that we did. We already talked about the external stuff like gaping and that type of thing. And there was a lot of things we could have picked from, but to simplify things, I chose to look at feeding, tissue disintegration, and digestive tubule atrophy. For digestive tubule atrophy, 0 is good, 4 is bad. And then you've got that range in between. So again, let's take a look at June. Not a lot of them are feeding, but you have very low tissue disintegration and really good digestive tube atrophy, or you don't have much of it, which means they're feeding. That's what that means. They're feeding and they're getting the nutrition into their tissues while they're feeding. We get a drop off in feeding at Gulf Jackson, about the same at Dog Island in July. But look at the condition of the digestive tubules. They're not in good condition now. They're moderate condition on average. Not quite a 4. 4 would be like death. But, you know, they're certainly not what they were in June. And you've got a lot of tissue disintegration going on. It could have been anywhere. I didn't distinguish between the digestive tract, the connective tissue, the meat. This is just in general. August, we look better in some places. We're feeding better now. Depends on where you are. The tissue degeneration, the atrophy is getting less. You're getting back to that 0. And then again by October, everything's good. And look at how many of them are feeding at that point, almost 100%. So they really do pick up feeding at that point. Whatever's out there, they're enjoying it and they're eating it. And then pathogens, these are the pathogens inside the tissue. So not the outside mantle, but the bacteria actually are infected with, I guess it's the pathogens that they're actually infected with. A lot of the clams had parasites. We're going to ignore that because it was very low, an average of 2 per section in the clam. That's not a problem. That's typical when you're out in an estuary. That's normal to have, even though it looks like a lot. It's a bacteria we want to take a look like inside. And it mirrors that bacterial vibrio population and that change in diversity that you see in the mantle in July goes up significantly. And your hemocyte infiltration or the hemocyte response, if you think back to that picture that I showed you with all the purple hemocytes in the swollen tissue. So the hemocytes are coming to the rescue. They find bacteria in the tissues and they're going to fight it, basically, is what you're seeing here. The bacteria starts to decrease in August. You've got a little bit of a lag or delay in the hemocytic response. They're still fighting what's there. And again, boom, October, we're back. We're good again. We're good again. So you've seen this from Leslie. You've seen it from Todd. You're going to see it again here from me. Basically, that's why I got this from Leslie. So this is just the average comparing from Dog Island to Gulf Jackson and what optimal should be. Again, you can see in July, you've got that highest temperature. It's almost always, well, it's average right at 90, but you can see where it peaks up over 90. Still fairly high in August, very good in October, but it's the same at both sides. There's really no difference. This is an interesting one to me, and I would have expected to see more from that. I know Todd had 2 as his, but to me, I don't like 2. 2's deadly.
Okay.
Under 2
Yeah, it
kills.
is.
So we don't want 2. What we want is optimal. We want around 5. 4 is acceptable. And you have a very different picture between Dog Island and Gulf Jackson. But honestly, it didn't seem to make a difference in the stuff that I reported to you as far as a health assessment. Kind of surprised me. But a lot of times you can get that 1-2 punch. High temperature and low dissolved oxygen are not a good combination. You can certainly see that in Dog Island and July, especially the lowest that you get here. Salinity, you know, Leslie reported a lot of variation, and you can see the variation just looking back at the arrow bars. But you know, all in all, if you just took a monthly average, it is within what we would call acceptable range for the most part. I didn't see, and salinity doesn't play much of a role in, well, it can play a role in parasitic pathogens, but it doesn't in bacteria. That's usually temperature. And interestingly enough, all that gaping that we saw is a typical response to temperature. That's a known environmental stress. Now, this next slide is going to look super busy. I'll just warn you right away.
That's cool.
But I tried to pull it all together. And again, I like the stoplight colors. So again, you can see everything that we talked about in July, all that red, which is bad. And you can still see it lingering in August, but it's getting better. And you can see how conditions are fairly equal in October to what they were in June. And then this is that survival that was talked about before. This is average across the different sites. And you can see, you know, how it really dived for both sites in August, but it was much worse at both Jackson and both sides again in October. That was just a continuation of all the problems that you saw. You know, you get that delayed effect in survival. And I didn't see a smoking gun as far as pathogens. So we can't really blame it on that. Temperature obviously played a role, you can tell that. But there has to be, and that's why I said I think we all need to get together and try to put the pieces of the puzzle together. What else besides temperature is involved? I'm assuming if there was a bacterial diversity change, there was probably a planktonic change. So there's your food.
Okay.
And bacteria, plankton are known to graze on bacteria. So that could have been responsible for the bacterial changeover diversity, perhaps what you had out there. Were there any nutrient changes that went along with this? That, you know, that we would increase in nitrogen or phosphorus or waste or anything else that, you know, I don't know. So that's it, and that wraps up my presentation.
So I want to just comment that this workshop is being recorded, and all the presentations as well will be put on my website so that you can review this later. I also wanted to say, and I didn't think I did that in the introduction, after the presentations, we have 2 more to go and we're way ahead of time, so don't start looking at your watches. We want to have a breakout session, so we want to then be able to go to each presenter and topic and get further input from you in terms of questions that haven't been answered, or what does this mean, what further research. And if you stay that long and get through that, you'll get a prize. We're also locking the doors. So you have to stay. Yeah, okay. So I didn't mean to cut you short. No, that's, if there's some brief
I didn't
questions.
see any questions in the chat.
Yeah.
Yes.
It's not a question, but it's a comment. What you taught me, I have to learn something. Yes, no, so it's not, it validated what I thought. Because I try not to, when I buy seed in the fall, I don't like purchasing seed before the end of September. And everything you just showed me validates why I do what I do.
Okay.
But my question is, I hate that it was only started in June.
I know.
Because if you would have did the whole year cycle, there's another month or another window that mimics this in the springtime of what October is. And if I would have loved to have known that mimic. And then you take from the October window where everything starts to trend up. If there would have been research done from the time the October window and then took it to May, because I think there's a peak there.
Yeah.
That we've never looked at, and I don't know.
I agree with you.
I'm saying what I'm saying is thank you, because you validated what I've tried to figure out. I knew what I was doing, but I didn't.
But you didn't know why.
Now I have confirmation.
So you know how we get a seed loss sometimes in April and early May? And we, you know, there's some things going on there that attribute to that, sometimes that almost routine little bit of seed hit.
It has changed for the simple fact is we have not got, you know, in April through March.
Yeah.
For years, and I'm talking for years, it was the whole growth thing. Beware the eyes of March, because you've got northwest winds and rain.
Yeah.
We don't get that anymore, and because we don't get that,
yeah,
we have not seen that mortality. I mean. In our seed production, because it used to be you would see this coming,
yeah,
and you know it's going to be here today, so you would go out there and you had a better chance of lifting everything up and moving it on the other side of the island to protect it away from the fresh water, because I mean you could go bass fish on Gulf Jackson, but not if that doesn't happen.
But,
So-
you know, you talk about trying to do this and extend this type of monitoring program, you know, the amount of work that Susan and her lab did was quite extensive. Yeah, I mean, the amount of man hours to get the histology results are amazing, to read those and have them available. And it's also cost a lot of money. We were able to really, take the little bit of funding we got from Florida Sea Grant and be able to spread it across to hit various topics that would affect clam mortality. And, this speaks to the fact that, you know, for ongoing research and monitoring, we need those funds. Much like you talked about, VIMS has ongoing funds allocated.
Well, I mean,
Require
I
those.
brought this up in another meeting. But we talked about how we tolerated test on clams. And then we're gonna take fruit stock from these clams. And, you know, that survived this heat, this thermal testing. Well, my question is, did we actually genetically modify that clam through this thermal testing? Or are we swinging in the dark?
No, you're not genetically modifying, you're just selecting with natural variation of genes, it's not
Exactly.
GMO, never say GMO.
Okay, but my question is, what is the difference between that and a regular flame that's not been thermal
You're selecting for certain
Traits.
Right.
Different genetics.
through, there's stuff, yeah.
It's genetics, but it's
Well,
not.
you know what, let's
genetically
do
modified.
because
Yeah.
I think we need to hear from Sarah. Hudson's here and she's gonna talk about her Hudson. And again, I should say that we're ahead of time, but we do want 5-minute discussion with each presenter at the end. So we can take home what's next, what's needed, or what other questions, come on, Sarah. So
See, it's.
that, here's your point.
Oh.
Yeah. If you kind of stand close to that.
Yeah,
can pick
everyone
that up.
hear me okay? Oh, okay. Hi, everyone. I'm Sarah Hutchins. I'm a staff scientist with the Gulf Shellfish Institute, located in Palmetto, Florida. And today I'm presenting on our work on assessing performance of thermally selected northern hard clams, so your Mercenaria, during the graph stage. So as a bit of an overview, as of 2023, for the Florida shellfish aquaculture industry ranked 3rd in the nation for production, which is mainly focused on hard clam production. But the industry as a whole, especially that hard clam production, is rapidly growing. And our production is focused primarily in and around Cedar Key, Florida. So in the summer of 2023, we all know we experienced the high clam summer mortality. And around that same time, we experienced these extreme summer temperatures. So specifically here, the week of August 8th in 2023, we had these really high temperatures, especially in these shallow coastal waters. This graph is specifically for Gulf Jackson, Florida. So GSI, we're actually able to look back at a 10-year data set from NOAA, their temperature in Gulf Jackson, looking specifically at these summer temperatures. And we were able to identify this week as a marine heat wave using this temperature data. You can see that 90th percentile of temperatures over that 10 years. And using the Hobday et al. Definition of marine heat wave, it's 5 or more days with sea temps above the 90th percentile. So we were able to confirm that this was a marine heat wave event. So a bit of GSI previous history. We ran a series of experiments looking at sublethal effects and lethal limits of northern and southern hard clams in response to essentially a marine heat wave, and also coupled with a low salinity event or hyposalinity. And what we saw was that both species, when they experienced heat stress alone, survival was great until they got above 35 degrees C, and then there was an extreme drop off in survival. And then when we coupled that high temperature stress with low salinity, we saw drastically lower survival in both species of clams. So we were actually able to use results from these experiments to then heat shock broodstock to start the selective breeding process. So we tried to naturally mimic the marine heat wave these clams naturally experience around Cedar Key. So we have a daily temperature, like a diurnal fluctuation in temperature. It's ramped up over 3 days and held over 4 days at that maximum set temperature. Here, we use the results from that experiment to stress the clamps at 37 C, so extremely high temperatures. And of the 100 clams we exposed to this heat stress, 28 survivors made it through day 7 of this marine heat wave. So we took these survivors, and with bay shellfish, we were able to condition and spawn these survivors. And then bay shellfish raised them to a 4 millimeter size. So moving on to this current study, we wanted to see if just this first generation, I'm referring to them as F1s, if they will show differences in survival and growth in these heat selected, thermally selected clams versus your conventional seed. So moving on to our timeline for this project, we outplanted these 4 millimeter heat selected seed at Gulf Jackson, Dog Island, both of those sites in June, as well as Tampa Bay in July. We stocked nursery bags at 10,000 seed each. And we also had some unselected seed planted alongside our selected seed at some of the sites where it's feasible. Unfortunately, we weren't able to do this at Gulf Jackson, but we did it at Dog Island and Tampa Bay. Although, no worries.
I was Gulf Jackson research.
So we tried to do those unselected seed where we could. We had a few issues getting those seeds to the farmers, and this seed we had at Dog Island and Tampa Bay, they either had slightly different density or timing with the outplanting, so it was difficult to get a true scientific comparison between those unselected and the selected. But we did go back and monitor everything 2 months post planting in August. At Gulf Jackson and Dog Islands, we stocked bags at like 1,100 seed for 10 millimeter bag. And then in Tampa Bay, they're stocked at still 10,000 in 5 millimeter bags. And then obviously we all know we experienced a significant hurricane. So we were unfortunately unable to go back and monitor the Cedar Key sites after this 2-month monitoring. So we actually focused our remaining monitoring efforts in Tampa Bay and went back for 4 months post planting in December and restocked these seed into 12 millimeter bags. I get disconnected.
Oh, sorry. Yeah. I think you.
Thanks, yeah. Okay, so our specific methods, when we outplanted these bags, we subsampled 6 bags at each site. 50 clams were subsampled per bag, and we measured shell length and whole wet weight. So we measured all 50 clams together, and then from that extrapolated to estimate an individual weight per clam. When we went back for the grow out bags, we counted survival and subsampled those same 6 bags at each site, and again measured shell length and whole wet weight. We also deployed those hobo conductivity loggers at when we outplanted at Gulf Jackson and Dog Islands. And we were able to recover the Dog Islands logger when we went back, but unfortunately the other one was lost to the hurricane. And then when we went back to restock the Tampa Bay grout bags, we again subsampled all the bags and measured, And we also took those samples back to the lab to analyze condition index, which is an indicator of overall health. So it's kind of general values. Higher condition index can indicate good health. So they're eating, they're producing gametes, spawn, things like that. And the lower condition will indicate, could indicate stress. So moving on to some of our results with Gulf Jackson. So on this figure on the left, we have the shell length in the dark blue and the weight in light blue. And you can see from outplanting in June to August, we do have a general increase in both your length and weight in these thermally selected seed. So they're looking good. They're growing. And then for survival, we saw a drop to about 47% survival, which seemed to be pretty much on par with the typical seed you're outplanting. Seems to be pretty much on par. So for moving on to Dog Island, the water quality, we were able to log through Tropical Storm Debbie, which I thought was cool. We were able to kind of fill that gap when the UF in situ logger had to be pulled. So you can see here, we were able to capture that storm, and you can see the drop in temperature and salinity over that week. That's good. So that's pretty cool. And then for Dog Island seed, the length and weight. So our unselected seed were actually longer and heavier, but we did those seed were planted a month earlier. So it's again, that comparison is kind of tricky. They were planted a month earlier, so they're going to be bigger. So when we actually look at their growth rates over time, we see that our thermally selected seed are actually growing faster. So as opposed to the controls, around 50% increase in length, our thermally selected are about 69% growth. And same thing with our weight. They're growing much faster than those selected seed. And then looking at survival, we weren't able to get replicate counts on this, unfortunately. We just had one sample per treatment per time period. So no error bars on these ones, but we did see a general drop in survival as you'd expect, slightly lower in the heat tolerant or the thermally selected seed, but still pretty similar to those unselected seed. And that survival was a little higher at Dog Island than at Gulf Jackson. So lastly, moving on to Tampa Bay, we actually saw a significantly larger seed in our thermally selected seed than the controls. So you can see over all 3 time points, you can see longer and heavier plant and are thermally selected. And for survival, the site actually had the highest survival is about 60% in August and pretty close to that in December. We didn't see a huge drop off in survival, which is pretty exciting. And the last thing we looked at was that condition index of these clams, which we did not find any differences in condition index. You can see this y-axis is pretty close, 1 to 1.5. So there's really not much of a difference between these two. But that could be because these clams were so small, so young, we may just not be seeing a treatment effect yet, or we might just not be picking up on an effect until the second generation. So moving on from this project, we actually were able to find those Dog Island clams. So the farmer reported there's about 80% of the bags still there, which is really exciting. We will be able to use the largest seed we have there and in Tampa Bay moving forward to produce F2s. So we are again going to repeat that heat shock and kind of expand on this project, make it more scientifically rigorous, and monitor the F2s, see if we'll see a stronger treatment effect in the second generation. And we're also going to, in addition to heat shock, we're going to have a set that we expose to that low salinity stress to see if we can create some high temperature and low salinity resistant or resilient hard clams. So we want to use this research to help support the Florida shellfish industry in becoming more resilient with these heat waves and freshwater intrusion that we're seeing consistently. And I want to thank the GSI staff for supporting, helping with this project, as well as all of our farmers, our project partners we have here, especially Bay Shellfish, DJ Struck, Joanne Clams, Joe Cannon, Heath Davis, and UF and Fort C. Grant for funding. Thank you.
You haven't seen available yet?
So we have seed at those 2 sites. So we don't know where they're going yet, but that's definitely a question to see if we can start distributing them. Since we're not seeing strong effects yet, we'll likely do these F2s and make those more readily available to farmers. So feel free to get my email address and reach out to me about obtaining seed.
Got anything that made the broodstock size yet?
So these F1s are still in grow stage, but the ones in Cedar Key, we're hoping they'll be ready in August. So yeah, I forgot to mention we have funding from ARC to create those F2s, and we're working with UF specifically Dr. Wei Ping Yang, and working on creating those F2s and getting the broodstock ready for that.
Right, so you want to participate in being a research model? Sure. That's, I mean, that's, yeah. They're far from doing the commercial staff service yet.
And the ARC funding, I think will be there July, and this will be the go around, and it should be to the farmers by the end of this next go around. So.
Yeah, so feel free to take a picture, email me if you want to help us out here. It's good.
All right. So do you think that this is just the way that we should go in Florida, is just to breed heat resistant stock and not even mess with the way it's being done now in the hatcheries?
No.
So I'm gonna tell you right now.
I'm asking, I'm putting it out there.
Do you know how many generations since 1993 have, and with really no rootstock selection program ongoing in any hatchery?
Yeah, there's no.
You know how many selections have occurred naturally where they get your harvestable clams and that's used for broodstock for the next generation? We have been doing heat selection for 30 years.
30
years.
Naturally.
Yeah, naturally. And I guarantee you, we have moved that.
Yeah. We're moving things on forward.
Yeah.
Yeah, we're just giving them the
but
extra this year.
that's long term. So now genetics has moved also far ahead. And I just, to complement the work that GSI is doing, UF also has a similar program and is part of the National Sea Grant Hard Clam Selective Breeding Hub. And those geneticists that are lead investigators out of Stony Brook and Rutgers University, they're the ones that have finalized the genome of the hard clam. So we know how it works in terms of genetic makeup. They've also just recently, through all the testing, have been able to identify those SNPs or genes that are related to heat tolerance. So yes, they do. It's not many, but that's where you start, because there's tons of SNPs, thousands of thousands. You can't afford to do that. Once they've identified that, you can start selecting.
Right.
So Dr. Hui Ping Yang leads that work under the hub. We do want to dedicate a seminar with her and her students to bring you up to date. They also are moving forward with F2 generation, but looking at it from a genetic perspective for selection. They also do heat shock. That's how you move forward. I think that Jamie.
Kurt has a question first.
Okay.
Go ahead, Kurt.
Oh, well, first of all, thank you all. It was a pleasure this is Kurt Hemmel with Bay Shellfish Company. It was a pleasure to work with the Gulf Shellfish Institute. One minor thing, mine and Nick's last name are misspelled, but I'll send you that. And but that's very minor. What I did want to say is also on the project, I wanted to note that there may be some potential, the research that was done the results could be more significant than they show, because one possibility is, Tampa Bay it is more difficult to grow clams in Tampa Bay. It has historically for, well over 2 decades. So, if that's general overall stressors, and then the results were amplified by using these stocks, the survival that is of these heat tolerated ones in Tampa Bay, that could be correct. Maybe it just wouldn't show as much in Cedar Key. So I do think it's relevant. I do think it also might point to the fact that this work should be continued, and I'm glad that you're doing it. Second thing would be, I'm glad Leslie mentioned something, because I have a comment and a question. My statement is, I heard Wei Ping present at ARC about the upcoming work, but I was part of the selection group along with Dan Solano and a whole host of people, that voted for Wei Ping to be added to UF as a researcher. And I am a little dismayed, and I'm a little, upset that we are, why haven't we seen the F1 generation that she claims to have done the exact same work? Why hasn't that been presented to us? Why? She said that she started her work in 2019, when I listened to the proposal just, a month ago. So that was my comment. Now my question, that is a question also. So Leslie, maybe you're the closest person to her in this. Can you?
We're here. We're Wei Ping's graduate students.
Okay. Well, can you potentially share any results from that F1 work that's been, put into Cedar Key? This is the right forum for it, and this is where we'd like to get the information.
Yeah, so we're going to be talking, it's me and Paul McDonald, another PhD student. So Paul? Yeah, you want to start?
Yeah, so we were able to test the F1s again with the heat shock. And we did see significant differences between actual heat shock selected animals and their control counterparts. Right now, we are in the process of creating those F2s for that group.
We want to verify the genes that we're looking at. It's not just like, oh, hey, straight up F1 is going to have, the significant genes that we're looking for, the significant SNPs. We have to verify it over a couple of generations. We can't just, look at the data and say, oh, this is significant, and then just cut out everything else. There are other significant genes. It's not limited to just one thing.
And we were able to do that with what's called a genome-wide association study. And pretty much what that is that we took a lot of certain markers inside the genome, and then tested to see the survivors, what they had compared to what the ones who died did not have. And so through that, we're slowly accumulating what is best way for genomic selection in the future.
Well, thank you very much for sharing that.
Okay. Kirk, to answer your question, we certainly are looking to a date in the near future to have Dr. Yang and her PhD students give these results to date, and how they're moving forward, and how we can parallel with the ongoing research done by GSI, which is, Okay, Joey?
I asked Mike because I've been interrupting. But what farmers is Wei Ping on?
Right now?
No, for this upcoming project.
Well, I don't know, but she will be. They'll be outplanted in various areas. I don't know, but And Dr. Yang's not here to answer, so I
No, here's no-
think we should move on.
No,
Yeah,
I just didn't know,
yeah.
I want to volunteer.
Oh, good, all right.
I'm not trying to back you, want to volunteer.
And we are trying to coordinate.
Write
with
that down, Paul.
this next ARC project, since we're working with them, we are trying to have a spawn at the same time too, so we can be out planning and looking at differences between their genetic selection and our selection.
Well, you have me already.
So.
That's it. Next up, Dr. Shirley Baker. I love this title. Breakfast. I'm that nervous. Yeah. Oh, sorry. And
I'm short.
yeah. And you have.
Yes.
Don't show.
Okay, so I'm Shirley Baker. I'm from Fisheries and Aquatic Sciences. And I'm gonna tell you about some of the work that I have done. So I don't need to show you guys this again. You've seen it like a zillion times now. It gets really hot, right? It's like getting up there to close to 95 degrees in some places. So that in warm water, clams have to work harder, doing all the things that they do. So what do? They ingest food. Okay. They ingest food, they digest, some of that food they just get rid of as pseudofeces. Some of it, it comes out as feces, but after digestion, they excrete nitrogenous wastes like we do. The energy that they assimilate goes to maintaining their bodies, doing all the things that their bodies need to do, moving and so on, growth, storing energy, and reproduction. And all of those things that they do require oxygen, just like we do. So how do they breathe? They have gills that collect the food. They also get oxygen flows across those gills. Here's where the mantle would be, Joey. And they have an open circulatory system, so they don't have closed vessels like we do, and red blood cells, but they do have cells, and they do have fluid that moves around their bodies. Okay. Now, one thing to remember is that clams are ectotherms. What does that mean? So we are endotherms. We make our own heat. We have body heat. Clams don't do that. So their body heat is always going to match the environment. So, if it gets hot out, hopefully we're maintaining our temperature the same as it always is. Clams, on the other hand, when the temperature goes up, their body temperature goes up too. And so why does that matter? That matters because their metabolism, so their oxygen uptake, their behavior, all those things are really tightly linked with water temperature. So even tiny increases in temperature means that all those things increase in rate. So their metabolism or oxygen uptake goes up with temperature, usually in a kind of a log rate. Okay. So when there's warm water, there's also low oxygen. So as temperature goes up, the oxygen content of the water goes down, but the clams need more oxygen when there's a higher temperature. So just when they need more oxygen, there's less of it, which is a problem. So that is going to result in stress, reduced growth, and so on. At some point, temperature becomes deadly. So they're increasing their, whatever they're doing, increases with temperature, but at some point, they're gonna crash. So what's happening then is that their proteins,
their enzymes that do all the work in their bodies physically start to unravel. And they can't repair those fast enough, waste build up, they use up their energy and so on. So there's some temperature at which things get really bad. So I wanted to set that stage for some things that we did last summer, but I also wanted to tell you about some things that we've been doing for a very long time. We've been doing all kinds of experiments. However, most of those, we never went above 89 degrees, but we know the temperatures get hotter than that. So for example, one of my students in 2007 looked at clam survival at 89 degrees. And they did fine for 21 days, as long as there was a normal amount of oxygen and salinity was 25 parts per thousand. If things were, like 10 parts per thousand, they died. If things were low oxygen, they died. But we have a lot of data. She also looked at burial behavior. So when there was normal oxygen and 25 parts per thousand at 89 degrees, most of the clams were burying themselves pretty quickly. That changed if, for example, there was low oxygen. And another student looked at oxygen uptake at different temperatures, or their metabolic rate, how fast they're doing all the things. So it, they're ectotherms, so it's low at 68 degrees and it goes up. But again, we only went to 89 degrees. So,
Sure.
Yes.
Wasn't that oysters on the last page? Diploids and triploids?
No, these were clams. We were doing diploids and triploids back then.
Oh, really?
We did. That was a long time ago. Yeah. And there wasn't any difference. That's why I just left it.
Actually, the triploids did worse.
Yeah. Okay, but we know that it gets hotter than 89 degrees. So this summer we did some additional work going up to 93 degrees. I wanted to go hotter, but I couldn't get my system to do it. So I talked to Sarah. Okay, so the oxygen uptake, or their metabolic rate, peaked at 88 degrees, but there was a lot of variability here, which suggests that some clams were performing really well. Some clams were probably starting to crash already. And then there was, on average, a lower oxygen uptake or metabolic rate at 93 degrees, indicating thermal stress and breakdown of normal physiological function. We also looked at clam behavior. This is actually similar to what Susan was doing. So we looked at gaping. So this isn't like gaping like I'm dead. This is gaping like you poke it and it doesn't really want to close. Highly extended siphons and just slow to react. So at 93 degrees, about 50% of the clams exhibited this abnormal behavior. And that would leave them vulnerable to predation, pathogens. I am assuming it would impair their feeding. I haven't looked at that specifically. And it's, it's an early warning that they're on their way out. We also looked at some clam mortality. Again, I wanted to go higher than 93 degrees, but I just couldn't get it.
Now that's worth doing.
But even after 18, these things wouldn't die. It was like Christmas, and I was still like, these things wouldn't die. Yeah, this is the ones that I did later in the, so there weren't any immediate lethal effects, but there were definitely kind of a chronic effect of heat So 93 degrees seems to be above their thermal optimum. And this extended exposure did lead to increased mortality. Okay, so kind of putting it all together, the metabolic performance, oxygen uptake peaks around 88 and then declines, suggesting that thermal stress starts around there, somewhere between 88 and 93. But there was a lot of individual variation, which means that we can.
This means that you still can select.
Yeah. So that's cool. The behavioral stress, gaping behavior increased above 90 degrees and over 50% of the clams gaped at 93. And then the cumulative mortality was 28 or 20% by day 18 at 93 degrees. And the delayed mortality indicates a cumulative effect of chronic heat exposure. So they can cope, but their long-term survival is compromised. So kind of the overall takeaway, 93 degrees seems to be a tipping point or somewhere between 88 and 93.
93 degrees Fahrenheit.
Yes.
Fahrenheit.
Wow, that would be cooking this. Yeah. Sorry, I tried to convert everything. I think in Celsius, tried to convert everything to Fahrenheit. Clearly didn't quite happen. But metabolic, behavioral, and survival data aligned to show a progressive breakdown under thermal stress. So I have lots of ideas about further research. We could do recovery after heat exposure. We could do the diurnal fluctuations, which are more realistic, right? I could collaborate with Sarah and do some of the selected clam lines. Individual monitoring, so looking at, well, if this clam is doing better, does it continue to do better? And then multi-stressor experiments, we know that when there's low oxygen or low salinity, it makes things much worse. But I mean, I could do all these wonderful things, but does it mean anything? Does it really? I want to do things that will contribute to helping you guys, not just things that I'm interested in. So I think that's what we'll be doing next. And that's it. And you're welcome to call me.
I agree. I agree. And I think what's really awesome is this was a very little amount of funding, disaster funding through Florida Sea Grant to support the number of topics that were addressed here and the folks that invest their time, the researchers here. I think is really a lot of output, but just points to the, again, the need to go further and support this type of work. You want to go to, so my intent, I'm always optimistic that I'll have more than 3 people at these workshops from industry. We got 4, I'm good. Okay. So I was planning these breakout sessions. We're not going to do this. We're not going to do this. What I'd like to do is just wrap this up. We're on time. I'd like to wrap up this session or this workshop. And instead of having you moving around the room, I'm going to move. And I want to get your final thoughts or additional questions for each of the topics that were presented to you today. What's future research efforts? And Joey, I know you're going to be let's talk about this when we get to her, to Dr. Baker's presentation. I would like to just review each presentation. What questions weren't answered? I know you had a question, I wasn't able to answer it. So let's get those questions out, but let's also help drive this future research. What would you like to see? And is there any potential management decisions? I'll get to that. For the folks that are on Zoom, I think the easiest thing to do and want your input is put it in your chat. We'll collect it. Or just send me an email, lnst@ufl.edu. We'll collate all of this. This will all be, again, posted on my website after this workshop. So let's just start with water. Is there any